Here's something to think about. Your brain can survive learning a new language, aging by decades, even certain kinds of injury, and its basic rhythms keep ticking along as if nothing happened. How does it pull that off?
I mean, I always assumed the brain was just really tough, but there must be something keeping it in line, right?
Exactly. Today we look at a study that simulates an entire cortical network and shows how a single, elegant plasticity rule keeps the brain's oscillations stable, no matter how you shake the parameters.
So let's start at the top. When researchers talk about the brain's functional connectivity, what do they actually mean?
Think of it like a phone network. Functional connectivity is a measure of how correlated the activity is between different brain regions over time, not whether they have a physical wire between them, but whether they tend to fire in coordinated patterns. If two regions consistently oscillate together or their signal envelopes rise and fall in sync, we say they are functionally connected.
And we can measure that from outside the skull.
With MEG magnetoencephalography, a technology that detects the tiny magnetic fields produced by electrical currents in the brain, yes. MEG is special because it can track these oscillations in real time at the millisecond scale. Unlike fMRI, which measures blood flow and is far too slow to see fast neural rhythms, MEG lets you watch the brain's electrical dance as it happens.
Okay, so the question is, what produces those coordinated patterns in the first place?
Right, and that's where large-scale biophysical models come in. Researchers build computer simulations of the whole brain, dozens of interconnected regions, each containing populations of model neurons, and try to reproduce the functional connectivity seen in real data. If the simulation matches real MEG recordings, you have a candidate explanation for the underlying mechanism.
But I'm guessing it's not that simple.
Not at all. The core problem is that these models are extraordinarily sensitive to their settings. Change the coupling strength between regions by just a small amount, and the predicted connectivity collapses or explodes into something unrealistic. Real brains don't work that way. They stay stable through learning, aging, even some forms of damage. So the model world and the real brain seem to live by different rules.
That gap is exactly what this paper is trying to close.
Precisely. The authors asked whether adding a specific biological mechanism, one the brain actually uses, could make the model robust in the way real brains are. The mechanism they focused on is called inhibitory synaptic plasticity, or ISP. Let me unpack both words. Inhibitory means the connections are between neurons that suppress the activity of their neighbors, not excite them. Synaptic plasticity means the strength of those connections can change over time based on experience.
So it's like a volume knob, but for the braking system.
That's a great analogy. In the brain, you have excitatory neurons that push activity up and inhibitory neurons that push it back down. ISP is a rule that adjusts how hard the inhibitory neurons press the brake, depending on whether excitation is running too hot or too cold.
And the brain does this automatically.
The idea is yes, the rule works like a thermostat. If excitatory activity in a region rises above a target level, local inhibitory connections grow stronger, more braking. If activity falls below the target, inhibition weakens, less braking. Over time, each brain region self-adjusts to hit its own activity target, without any global coordinator telling it what to do.
Wait, so every region is tuning its own brakes independently.
Exactly, and that's the key. Because different brain regions receive different amounts of long-range excitatory input from the rest of the network, they each end up with a different inhibitory strength. Regions that are heavily connected, getting lots of excitatory drive from far away, develop stronger local inhibition to compensate. Quiet regions need less. ISP produces this heterogeneity automatically.
That sounds almost too elegant. Why hadn't models included this before?
It had been demonstrated in simulations running on the slow timescale of fMRI, where the dynamics are relatively simple. But at the fast timescale of MEG, where oscillations are happening around 11 times per second in what neuroscientists call the alpha band, the concern was that these rapid oscillations might interfere with the slow plasticity process. The authors were the first to rigorously test whether ISP actually works under these fast, noisy, oscillatory conditions.
So how did they actually set this up?
They used a model called the Wilson-Cowan neural mass model, a well-established framework originally developed in the 1970s. Instead of simulating individual neurons, it treats each brain region as a mass, two interacting populations, one excitatory and one inhibitory, and describes how their average firing rates evolve over time. The key strength is that you can scale this up to a whole brain relatively easily.
How many regions did they simulate?
68 cortical regions, based on a standard anatomical parcellation called the de Zinckenkielny atlas, which divides the cortex into gyral-based areas across both hemispheres. Each region had its excitatory and inhibitory populations connected locally, and the excitatory populations were linked across regions by long-range white matter connections.
And where did they get the map of those connections?
From diffusion MRI data, a brain imaging technique that traces the direction water molecules move along white matter fibers, effectively mapping the physical cables. They used data from the Human Connectome project, averaged across 40 subjects, to build a 68 by 68 connectivity matrix. They also incorporated distance-dependent propagation delays, because signals traveling longer paths arrive later. And at MEG timescales, those delays are on the same order as the oscillation period itself.
And the ISP rule itself, how does it plug into the simulation?
The inhibitory connection strength in each region changes slowly over time according to a simple product, the local inhibitory activity multiplied by the difference between local excitatory activity and the target level. If excitation is above target, the inhibitory strength grows. If below, it shrinks. The learning rate is deliberately set very slow enough that the plasticity does not interfere with the fast oscillations. The two processes operate on separated timescales, like a thermostat operating on hours while the furnace cycles on minutes.
How long did they actually run the simulation?
1500 seconds, about 25 minutes, to let ISP fully converge. Then they switched ISP off and ran another 500 seconds to measure the stable functional connectivity of the resulting balanced network. They compared this against 3 connectivity metrics computed from MEG data recorded from 55 healthy participants.
3 metrics, tell me what those are.
The first is amplitude envelope correlation, a measure of whether the overall strength of oscillations in 2 regions rises and falls together over time. The second is the phase locking value, which captures how consistently 2 regions maintain the same phase relationship cycle after cycle. The third is the phase lag index, which quantifies whether one region consistently leads the other in phase. Each captures a different aspect of synchrony, and each has different sensitivity to measurement artifacts.
So passing all 3 simultaneously is a much tougher test than passing just one.
Exactly right. So what did they find? Without ISP, the model's match to real MEG data was decent, but only within a razor-thin slice of coupling strength values. Nudge the global coupling up or down even slightly, and the match collapsed.
Like trying to balance a pencil on its tip.
That exact image, with ISP switched on, that needle-on-a-point balance transformed into something much more like a table, stable across a wide range of coupling strengths. The range of parameter space where the model reproduced realistic MEG connectivity expanded dramatically.
And all 3 connectivity measures improved simultaneously.
That's the headline result. Without ISP, the optimal parameter point was different for each metric. You couldn't find a single setting that was good for amplitude envelope correlation, phase locking value, and phase lag index at the same time. With ISP, all 3 metrics aligned at the same operating point. One set of parameters, all 3 measures of connectivity looking realistic at once.
How did they know realistic actually meant something? Wasn't it just relative?
Sharp question. They used individual human variability as the benchmark. They computed how similar each of the 55 real subjects' functional connectivity was to the group average. That gives you a natural ceiling, the best any single subject estimate can typically achieve. Then they asked whether the model performed significantly worse than a real person. The answer was no. The model's similarity to the group average was statistically indistinguishable from typical human-to-human similarity in all three metrics.
So the model is performing about as well as a real human brain.
Within those terms, yes. And that's a meaningful bar, because the model was using a single average connectome rather than a real individual's anatomy.
What about the excitation-inhibition balance itself? Did ISB actually achieve it?
They tested this directly. After ISB converged, they examined each region's local inhibitory strength and compared it to that region's total long-range excitatory input, essentially how strongly connected it was to the rest of the brain. The correlation between those two quantities was high. Regions with more long-range excitation had developed stronger local inhibition to compensate. The relationship explained roughly 65% of the variance across regions. The balance was real and spatially organized, not random.
That's remarkably clean for a self-organizing process.
It is, and it emerged most clearly in the metastable regime, the interesting middle ground where the overall synchrony of the network fluctuates in time rather than being locked at a constant high or low value. Metastability is also the regime that produces realistic functional connectivity. All three features, E/I balance, metastability, and realistic MEG connectivity, coincided in the same region of parameter space.
So what's the actual mechanism? Why does balancing excitation and inhibition make the model more robust?
Think of it this way. In the unbalanced model, each region's behavior depends heavily on how much long-range excitatory drive it happens to receive from the rest of the network. Regions that get a lot become overactive, quiet regions stay subdued. The whole network's dynamics then hinge sensitively on the exact global coupling strength. Too weak and the network is silent, too strong and it saturates. ISB equalizes activity levels across all regions by adjusting local inhibition to each region's unique position in the network. Once those activity levels are equalized, the network's behavior becomes far less dependent on the exact coupling value.
It's like putting individual volume controls in every speaker in a concert hall, so the overall sound is even no matter where you sit.
That's a perfect analogy, and the consequence for synchrony dynamics is elegant too. Without ISB, the Wilson-Cowan network showed strange, multi-valued behavior, adding more coupling first raised synchrony, then caused it to collapse, then raised it again in a qualitatively different regime where some regions were stuck in a high-activity steady state. With ISB, that messy behavior disappears. You recover a clean pattern, low synchrony with weak coupling, high synchrony with strong coupling and short delays, and a metastable middle ground in between. The same picture seen with much simpler oscillator models like the Kuramoto model.
They also tested a simpler version, using one uniform inhibition value for the whole brain instead of local ISB. What happened?
The global balance was still better than no regulation at all. For the phase-based metrics, performance was actually comparable to full ISB, but amplitude envelope correlation, which tends to be the most reliable and reproducible measure, was somewhat worse with uniform inhibition. So the local, region-by-region adjustment does add something. That said, the main driver of robustness appears to be getting the global balance of excitation and inhibition roughly right, not the fine spatial detail of each individual inhibitory weight.
What does that tell us about the real brain?
The authors suggest it means that robust dynamics may not require a perfect synapse-by-synapse tuning mechanism. A rougher homeostatic process operating at a larger spatial scale might achieve most of the benefit. The brain almost certainly has multiple plasticity mechanisms working at different scales and timescales. ISB is one candidate, but changes in intrinsic neuronal excitability and interactions with excitatory plasticity are also in the picture. The study's core message is that whatever the biological implementation turns out to be, the key is achieving that overall balance between excitation and inhibition, and doing so dynamically as the brain changes over time.
If I had to put today's surprise in one sentence, a single self-adjusting braking rule turned a brittle, parameter-sensitive brain model into something as stable as a real person's neural activity.
That's exactly it, and the implications reach beyond modeling. If ISP is how the real brain maintains balance through aging and learning, then disruptions to this mechanism could underlie conditions where that balance breaks down, including certain neurological and psychiatric disorders the authors mention as future directions.
They also want to test whether ISB can restore functional connectivity after simulated lesions.
Yes, that's high on their agenda. The preliminary evidence from slower fMRI-based models suggests it can, and this faster MEG timescale framework is the right place to put that to the test properly. The story is far from finished, but today's chapter is a compelling one.
Thank you so much. This was genuinely beautiful computational neuroscience.
The credit goes to Romesh Abissaria and colleagues at Oxford and Nottingham. The paper is a biophysical model of dynamic balancing of excitation and inhibition in fast oscillatory large-scale networks, published in PLOS Computational Biology in February 2018. Thanks for listening to OSCI Pod.